I don't know about France. I think he wants to hear about some. So it's right.
Yeah, there's not so much in France, actually. That's right. There's more in Spain, right, which is a much smaller company, right?
I didn't know about Spain. What companies are there?
Well, more researchers companies. They're doing research with other folks, COO, which is sort of their it's kind of oncology related, but they sell again space there. They built a nice cluster there. It's run with their Caltech, which we went to California. So some of the first work program where I thought some of this around on that. Yeah.
Do you need help with the Zoom?
I actually set up a YouTube live stream, so I think it's good.
Oh no. Those are the theorists at CERN that do that. I prefer less so of the tedious calculations, more so of the nice little mathematics activities. These are the phenomenologists that calculate
Right. It's more subtle science.
It's important.
Yeah.
People that take is this?
This is OBS, which is like a recording platform, and you can connect to YouTube and everything. So I don't think it's working right now, so we'll just stick to YouTube. We've already got somebody. Oh, 2 people are online. So hi guys, welcome. We'll be starting the meeting very soon. There's also live commenting, so if you want to ask any questions, feel free to just use the chat. I think it's there. And yeah, or just Slack me as well. All right, I think that we can get started. First of all, welcome. And thank you so much for coming to today's very special event with a world-recognized guest, Michael Ringel. Here at the Caltech Longevity Club, we are building a community of talented scientists and engineers who are driven to extend human health and lifespan. My name is Andrea Olsen. I am the founder of the Caltech Longevity Club, and I would like to invite you to join us on our mission of building the next generation of young longevity innovators who will be advancing the frontier of aging research. We're truly lucky to have such a special guest joining us today, Michael Ringel. And Michael is a recognized leader with decades of experience that powered innovation and strategy in the biotech and pharmaceutical sectors. He has been a key opinion leader in the fast-growing aging and health span sectors. And before becoming the Chief Operating Officer of Life Biosciences, Dr. Ringel served as the Managing Director and Senior Partner at Boston Consulting Group, BCG. He advises both biotech and pharmaceutical clients on portfolio optimization, research and development strategy, and organizational design. And Dr. Ringel has extensive experience in licensing, corporate strategy, and partnerships. He serves on the boards of Hevolution Foundation in the US and the American Federation for Aging Research. And Dr. Ringel completed his Bachelor's of Arts in Biology at Princeton, obtained his PhD in Biology from Imperial College, and a Juris Doctor from Harvard Law School. Please welcome Michael.
Thank you, Andrea, for that warm introduction, but also for all that you do to build a community here. It's really important. Caltech is a powerhouse, and building a longevity community here is going to help the field tremendously. So thank you all for being here, and thank you for Andrea for organizing it. So I'm going to talk a little bit about why this is such a good bet if you're interested for your career. Why it's such a good bet for society, and why I made a bet myself, why I put it with my feet. So I was at BCG for 25 years. I was a senior partner managing director, as Andrea mentioned, and I became so enamored of the technology that I jumped to a small startup biotech. I've been now for 4 months, and I'll tell you a little more about it. First, a disclaimer: this is a company that, I'm not speaking necessarily on behalf of Life, but just to make sure that everyone is aware that there are forward-looking statements. Not everything that I say is necessarily going to come true. There are 5 things that I want to talk to you about, and it's usually good to have just a list of 3, but I couldn't fit it in, so 5 total. One is just how important longevity science is if we want to make improvements in health and in lifespan. The second is that we need action at every step, and I'll explain what I mean by each of those steps. The third is the tremendous benefit here. So there's obviously health benefit, obviously lifespan benefit. Many of us believe that's a moral imperative in and of itself, and that's all you need. But on top of that, there is a tremendous actually economic imperative, and the companies that realize that economic value are going to bring value to society and also to themselves. So I'll spend a little bit of time on the financial side of this. And then I'll talk about my own choice around partial epigenetic programming, and then what you can do if you want to get more involved. So first, so why does this matter? So this is a chart of cohort survival, starting at 100% going down to 0 with chronological age. It's called a Kaplan-Meier curve. It doesn't necessarily need a special name. And what you can see are curves at 4 different time points in history. You can see actually there's been a tremendous improvement in how this curve looks over the last 150 years. And that has led to tremendous increases in average lifespan. Those were primarily through better sanitation, vaccination, and then better infant mortality care prevention. So if you look at the 1860 curve, for instance, it's fairly linear throughout lifespan, but there's obviously that relatively steep section in the first few years, which is largely non-existent in the developed world in the current curve. But when you look at this, you should also notice 2 kind of obvious constraints that we're hitting. The first is the curve has become more and more square with time, but at some point you can't make it any more square, and we're getting actually close to that limit. And the second obvious point is that despite those advances in the shape of the curve, the maximum really hasn't changed. That's true no matter what you spend. So this chart shows healthcare expenditure per capita on the x-axis, and the y-axis is health adjusted life expectancy, similar to overall life expectancy, but here a year in partial health only counts as a year of life, a partial year of life. Each dot is a country, and you can see there's an asymptote, right? Spending more is not necessarily getting you a better outcome in terms of health adjusted life expectancy. And whenever you see an asymptote, that sort of begs the question: are you running up against a limit? Which is the right question to ask when you see this. And the answer is yes. So some folks wrote in Science in 1990, looking at the data that I just showed you, and said, we're going to no longer have increases in average lifespan, which we have been used to in the developed world. That's going to stop. They made that prediction in 1990. They took a lot of flack for it, but it's really stood the test of time. So they said, based on the average lifespan that existed in 1985, which is around 80, if we want to get to 85, if we want to get to 90, if we want to get to 105, how much would we have to reduce the rate of mortality? You can see it was 55%, 70%, 90%. And that's in the context where if you cured all major degenerative disease, you would only reduce it by 75%. And so they said, we're at the limit of what we can do with existing methods. And so the quote on the right-hand side, the period of rapid increase in life expectancy in developed nations has come to an end. And they were correct. In fact, it's actually regressed in the US since this time. But that's not the full quote. They actually said unless there's a new technology that allows us to think about manipulating lifespan, manipulating the rate of aging. And they also say at the bottom that they strongly suspect, they're optimistic that this is forthcoming. So is there a reason for that optimism? So the way to think about that is the following. So everybody knows this, but it bears repeating so you think about it. Most diseases are diseases of aging. Actually, more than 90% of what kills us in the developed world are diseases of aging. You can see here cardiovascular disease, cancer, dementias, diabetes. These are really the scourges of humanity. They're all going up with age, and they're actually going up more or less exponentially. We all know that. We haven't typically thought of age as an intervention point to ameliorate disease. But if in fact it's possible to decouple biological age from chronological age and manipulate where you are on this x-axis, either slow the rate at which it progresses or even potentially move it back to the left, then it becomes a single intervention point that can address many diseases simultaneously. This is called the geroscience hypothesis. So is there any reason to believe that? So there's actually quite a lot of reasons. There's a lot of evidence that lifespan and the rate of aging, and therefore the rate of incidence of disease, is an evolved, genetically regulated trait, which means it is manipulable. So the first line of evidence is just theory, right? So energy and resources are not infinite. So what an organism can accumulate is limited. The organism has to do work with that limited amount of resources and energy that it can accumulate. So there has to be an optimal allocation of how it deploys that work into things like growth and reproduction on one hand, and maintenance repair on the other. And what's setting that optimum is not lifespan, but fitness, the evolutionary concept of fitness, which is looking out into some long-run future from now. How many copies get created? Or it's another way of saying what we see today are just those lines that were best able to proliferate in the past. And that's a function, yes, of survival to reproduce, but also number of offspring, time to produce offspring, and quality of the offspring, where quality is a recursive function on these other conditions. And if you run the math on this, you find that the optimal allocation is almost never in maintenance repair such that the soma doesn't degrade with time. And what sets the right optimum, it does vary by species, is the more likely the soma, which is the body, is to be destroyed by some extrinsic mechanism, to get eaten by a predator, to die from famine, to succumb to bad weather or pathogens. The more likely that is to happen, the less you want to invest in in maintenance repair of the soma, and the more you invest in the reproductive rate. So mice, for example, which die very quickly from these other extrinsic mortality conditions, and that's very little in maintenance repair. They actually get cancer in 2 years. Humans don't get cancer in 2 years, but they're not repairing those defects that cause cancer. And I'm starting to bleed into the second piece of evidence, which is comparative biology. So we can look at different organisms and see widely different lifespans, factors of 100 across vertebrates. Even in a closely related clade, so the colorful diagram there at the top are the Pacific rockfishes. They only diverged 8 million years ago, so it's a very recent clade. And yet some organisms, some species in that clade live 10 years, and others live 200 years. In a way that we can predict, those that are less susceptible to extrinsic mortality are the ones that live 200 years. They're bigger, so they have less predation, and they live at depth where resource variability is not an issue. That's across species. We also see it within species. The eusocial insects, these are ants, bees, wasps. There's some others as well, but those are the key ones. With the sisters with the exact same genes, or nearly the exact same genes, if you turn on certain ones, when it becomes a queen, live extremely long, extraordinarily. So queens of the common black ant live 30 years, whereas worker ants live 8 months, just by which genes have been turned on and off. We see it in plasticity based on conditions, so kind of pretty much any basic insult we get will actually make organisms live longer, which seems counterintuitive. The classic one is caloric restriction, so the graph at the top is Clive McKay's work at Cornell in the 30s, showing that as he starved rats, they lived longer. Not only do they live longer, but they're healthier, so the diagram on the bottom are 2 rhesus monkeys. The one on the top has been fed more than once. The one on the bottom is the same age, but has been semi-starved. Not necessarily something you want to not fun, but harks back to this first column. Like why would an organism, when it's suffering an insult, live longer? Because in conditions where there's a famine, it does not make sense to breed. The offspring are just going to die anyway. So that energy is redeployed into maintenance and repair, wait in those seasons. So a mouse in a caloric restriction lives 3 years rather than 2, and that's long enough to have one more breeding opportunity where the following year might be a good one. It's not just non-humans, we see this in humans as well. So the top is a graph from Phelan and Rose showing an inverse log association between caloric intake and lifespan among different populations of Japanese. Pretty much all the blue zones, Okinawa is one of them, have very low caloric intakes. The bottom is a health span data. This person is what's called a crony. Practitioner of caloric restriction, optimal nutrition, before and after. And many of these biomarkers are healthier as well. We see it in genetic variants. So kind of the paper that broke the field open was the paper on tops and Kenyan working in worms, found a single gene that makes worms live twice as long. Happens to be in the exact same pathway that makes this choice around allocation. And we see it also in humans. The chart on the bottom is that centenarians are enriched for variants in the same pathway, GF1. And the last one we see with pharmacological intervention. So there are drugs that you can give to other species, in this case mice, that make them live longer. This was work done at the National Institutes of Aging. It's called the Intervention Testing Program. This is the first molecule that showed this extension, but there are many others discovered since. So the only thing on this page that's missing actually is the bottom right, which is proof of a pharmacological intervention in humans, and that's just because it takes a very long time to run a lifespan trial in humans. It hasn't been. But I'm pretty convinced based on the other data that we would see an effect. So how do we tap into this? So that there's kind of a general theme in pharma development that if you want to make a drug, you need to tap into existing biology, whether that's the target you go after or whether it's the modality, the chemical tool you use to go after that target. So just some quick examples on that. On the left, this is for hypertension. If you want to reduce blood pressure, which is hypertension is high blood pressure, you hit something that's in the pathway that modulates the diameter of the blood vessel, right? Restricting and allowing it to relax, reduce the pressure, whether that's an angiotensin 2 receptor blocker or an ACE inhibitor, etc. You can reduce the volume of blood that goes through, right? Through either a diuretic that reduces the fluid volume or reduces the heart rate, etc. And on the right-hand side, the list of modalities essentially all tap into some existing biology, chemical mechanism that exists in the body to create a drug. So if we then think about that for drugs in this space, there are two kind of big areas to think about. The one on the left is what I was just describing around this switch that allows you to switch between conditions are abundant and conditions are restricted. There's a particular model called mTORC1 that modulates that. If you hit that pharmacologically, which Dr. Myson in the chart I showed you in mice extends their lifespan, it should potentially something like that should work in humans. There's lots of other targets that are related to that, both upstream and downstream. And on the right-hand side, I'm going to return to this in more detail, but there is biology built within us that make babies young from our older bodies. We're doing this, in this room, if you go and have a baby, that baby will be born an infant, even though you yourself are not an infant. And the more you think about that, the more you realize there's some profound biology built into us that allows it to happen. If we can tap into that, that's an opportunity. Okay, I'll come back to that in a bit more detail. But so the next thing I want to talk about a little bit is just social action and how important that is. So if you work backwards from wanting to have medicines of this kind on the market, what do you need? Well, first, working backwards, you need pharma companies, because most drugs come through pharma companies. There are a few that are done without their involvement in rare diseases, but pretty much if you're talking about treating aging, treating conditions of aging that affect all of us or many of us, it's likely to involve pharma. But about 80% of the molecules that pharma develops come from biotechs. So we need biotech involvement. They get most of their ideas from academics who rely on funding from places like the NIH to do their work, which is, I think, on everyone's minds right now. Those funding bodies are obviously responsive to legislative priorities in terms of where and how much. And legislators are, at least in the long run, responsive to public opinion. And so if we think about how we want to get action, we really need it on all these steps, but particularly the first one, to get people's minds around this topic and how important it is. And just as an anecdote, an example of that, I'll talk about the mind shift change that happened in cancer. Due to the work really of one person, largely, named Mary Lasker, her husband, the Lasker Prize is named after her. She ran a public awareness campaign to get people concerned about cancer. So it was this is kind of before all our times, including mine. Cancer was a taboo subject in the 40s and 50s, and she worked hard to make it not a taboo subject. You can see public opinion changing by the 60s. That public opinion led to policy action. So we had the War on Cancer, the National Cancer Act of 1971. That led to funding through the NIH. That led to a step change in academic activity. You can see starting right around 1970. That led to, a decade later, 30 medicines, and now our cancer mortality rates are down a third. So it is possible to do this, but it does start with people being concerned about it and wanting to have some action in the space. So where are we? So I'll compare geroscience to cancer. It's not to say that we should be doing less work in cancer. It's obviously an important field of research, but to give you just a barometer to measure things by. Again, working backwards, there's something like 500 medicines on the market for cancer. There are no approved medicines for geroscience. There may be some medicines that are actually working for this, like the GLP-1s, but they weren't approved that way. It's something like 60 billion in OpEx that pharma spends on cancer, much less than that for geroscience. Something like 5,000 startups in cancer, maybe 200 in geroscience. Something like 170,000 publications a year on cancer, maybe 10,000 in geroscience. You can see billions in funding versus less than a billion. A lot of high-profile laws around it versus actually nothing yet so far. And a real difference in public concern and awareness. So cancer, if you ask people, it's the number one or number two health concern they raise. Only about 5% of people, it's a little bit old data for 2011, but only about 5% of people say they have high awareness of this space at all. And they're decidedly mixed in how they feel about it. So that's that 5% on top. 5% say they've heard a lot about it. And then, do you think this was a good idea? Would you want medical treatments in slow aging? Would these be good for society? It's really quite split. And if you ask why, some of the concerns that are raised are on the right-hand side. Want this to lead to overpopulation, want this bad financial burden, want to prolong ill health, do we have concerns about equity? Obviously, these are all important concerns, but they're actually quite misled. So, for instance, in overpopulation, the longest lived societies have the lowest birth rates and actually declining populations. So, at least the empirical evidence is that this reduced populations, not increasing. On access, there's the saying, an ounce of prevention is worth a pound of cure. These small interventions that modulate the rate of aging may be much easier to monetize than late-stage cancer therapies that are hundreds of thousands of dollars per patient. But the one I'm going to dive in on is the financial concern, just to give a little bit more data on it, because it's so completely wrong. So why might people think that this would cause a financial burden? I think it's two things. One is on the left-hand side, which is a belief that an aging society drives healthcare cost and other entitlement spend. And what you see in the black line is increasing population that's elderly and an association with increase in entitlement spend, particularly the part of green, which is healthcare spend. Now, on the right-hand side, you have a kind of negative correlation between the amount of population that's over 65 or 60 in this case, and GDP per capita growth in that. So not only are they driving more healthcare spend, but maybe they're producing less as well. But this is a flawed understanding. A lot of serious economists have looked at this, and they found exactly the opposite. So starting with Murphy and Topol at the University of Chicago, who wrote a National Bureau of Economic Research paper that looked at gains that have already been achieved by people living longer from 1970 to 2000, and found that it was worth trillions of dollars a year. Danny Goldman at USC, using a model from Rand Corporation, assumed a very small change to the mortality curve. You can almost not see it in this chart. And phased it in very slowly, and found that was worth trillions of dollars. And Andrew Scott at London Business School made a kind of a different assumption and again showed trillions of dollars. And the reason why there's trillions of dollars here is that by slowing the rate of aging, you're actually delaying the time in which people have to draw down benefits. So typically shortening. You're also prolonging the period of time that they're contributing to the economy, whether that's through work or acting as consumers and so on. So it's actually the end, rather than an exacerbator of a regular wave, sort of 180 degrees of where people's kind of naive view often is. Okay, yeah. And actually, yeah, please, I'm almost done, but please, as I keep going, if there are other questions, just shout out.
Yeah, so I'm just curious, so are these three that you showed, are those conclusions dependent on the assumption that people actually have to delay their retirement to make that conclusion?
So just, I'm not sure I remember, but so is the conclusion dependent on the assumption you have to delay retirement to achieve it? So one of the drivers is the extended period of working. But if you set that zero, right? And by the way, it's not forcing people necessarily. You can still keep the retirement age as it is. Some people, including me, will want to work past the retirement age. The ability to do that voluntarily is not mandated, but would still, presumably come out through this. But even if you set that to zero, active as consumers is a driver, and then the delay and shortening of the period that you're drawing down healthcare costs, things like that, is a driver. There's also value of life itself, which you might say is infinite, but typically economists use what's called value of statistical life and use that in their calculations as well. And so there's some value ascribed to being able to live another year, which might be modulated by the quality of that year, but is still something greater than zero. It's a great question. Okay, so partial epigenetic reprogramming, what is that? Why is it the coolest thing since sliced bread? So the way to think about this is all of us in this room started out as a single cell. A single cell that had no identity. It's called pluripotent. It can be anything. And then as that cell divided, different lineages developed from that. So some cells become nerve cells, some become muscle cells, some become skin cells, etc. And so cells gain identity during this period of development. And then as we age, cells gradually lose identity. The marks that determine their identity get corrupted. And that is associated with loss of function, increasing tumors. And yet there is this amazing magic biology within us that allows us to reproduce and make a cell that is all the way back to that pluripotent, completely unaged cell from our old cells. And Shinya Manaka won the Nobel Prize in 2012 for showing how to do this through cloning. The application of just 4 factors, 4 transcription factors, small proteins that bind unit, abbreviated OSKM. And people were astounded by the pluripotency that you could take any cell and turn it back to a blank slate cell that then could become another kind. But what was sort of missed was that also when you do that, those cells were completely de-aged. So Dolly the sheep, which was a cloned mammal, was born as a baby, even though it came from an adult cell. And we now know we can do this in series over and over again. So you can take a mouse, take a skin cell from that mouse, clone it, starts out as a baby, grow that to adulthood, take a skin cell from that adult mouse, reclone it, starts as a baby at least 25 times in series. And the only reason why we don't have longer is there just hasn't enough time since the discovery to keep doing it. That's profound, right? So there's biology built within us, and we already know how to tap into it to actually completely de-age cells. Now, the problem is you can't do that in vivo in your own body, because you want your cells to maybe de-age, but not to lose their identity. If your heart cells stop being heart cells, I think that's a problem, right? There's also a cancer risk. But the insight that was on the cover of Nature in 2020 is that if you just treat with 3 of the 4 factors, OSK in this case, you can get de-aging at the cellular level, at the tissue level, and the organism level without getting all the way back to pluripotency. NANOC is a key marker of pluripotency. They saw the NANOC, they saw the tumor formation. And this is work out of David Sinclair's lab at Harvard. Another way to kind of see this is, many biologists in the room, I assume there's some people who are not biologists, there's something called the central dogma. And DNA is transcribed to RNA, which is translated to proteins, which then catalyzes metabolites that are within our cell. We can look at the central dogma again, working backwards, thinking about different cell types, skin versus muscle versus liver, as well as different cell ages, old versus young. So different cell types have different metabolisms. Young and old cells have different metabolisms. They have different proteomes, they have different transcriptomes, but they have the same DNA. So every skin cell in your body has the same DNA as every heart cell in your body, as every brain cell in your body, even though those cells look incredibly different, do different things. And the way that they're told to do those different things is what's called the epigenome. One of the key things are just little methyl groups on the cytosines that tell it which genes to turn on and off. And looking at this diagram, it's a pretty good inference, especially now we have empirical data, to say it's actually the thing that distinguishes between old and young cells as well. And what happens when you have an embryo and it goes through a period of genesis, actually those methyl marks are erased also supporting what we'll call the hypothesis, the insight here. And so that becomes an intervention point to say, if I can manipulate the epigenetic marks, these methyl marks, and I'll have a tool that allows me to de-age cells as well as obviously change identity. So that's what we're working on, building off that Nature paper. The platform is OSK, just 3 of the 4 novel factors that should apply in kind of every age-related disease that they can get delivery. But of course, we have to start somewhere. So we're starting with optic neuropathies, which are diseases like glaucoma. So glaucoma, number 1 cause of blindness in people over 60. I think most people are kind of familiar with it, or at least heard of it. The one on the right-hand side is NION. It's actually the most common acute optic neuropathy in people over 50. It's basically a stroke that damages the optic nerve. So you wake up one day and one of your eyes is not working. And you go to the doctor and they say, well, there's actually nothing we can do for you. There's no treatment. And by the way, it's about 30% likely to happen in the other eye over the next 5 years. So people with this really live in fear. It's quite debilitating when it onsets at that age. The technology is called a dual vector AAV. So it's basically a virus that you hijack to deliver a gene. The dual vector allows you to turn it on and off in the presence of doxycycline, which is a common antibiotic. So on the right-hand side, you can see the absence at the top, on the right, and the absence of doxycycline. There are no green dots or blue dots. In the presence, you get expression of the proteins of interest here, which are the things that light up in the bottom right. It was shown in the Nature paper to work in multiple models of mice, whether that was crushed, like physically crushing the optic nerve, whether it's glaucoma simulated through microbeads, or whether it's just eyes work less well as they age. In all of these, you saw improvements in function, improvements in tissue, improvements in natural vision. Also, safety data. I spent too much time on this, but they saw no markers of potency, no tumors. At Life, we've now recapitulated this in non-human primates. So mice are not humans. Neither are non-human primates. Monkeys, but they're closer. They have peripheral vision. They have limiting membrane. So more likely to translate for sure. We did it 2 ways. We did a prevention model where we started treatment with OSK before the damage, and then a rescue model where we started treatment only after the damage. The damage in this case was the lasering the retina. And what we found in both the mice and non-human primates is kind of the full logic chain we want to see to say this is going to work in humans, which is first we can see that we're delivering it to the right cells. The brown dots are expression of OSK in the retinal ganglion cells. We see the methylation pattern revert back to almost baseline. So the yellow and blue is what's methylated and demethylated before damage, in the middle after damage, but before treatment, and in the far right, the yellow and blue is after treatment, goes back to baseline. We see it in cell function. What's there is it's called pattern energy. It's the electrical impulse the nerve can generate. Starts out looking like the blue curve. If you damage it, it looks like the green curve, and then if you treat it, it goes back to the red curve, which is very similar to the blue curve. We see the tissue health. So literally the axon, the optic nerve, is regrowing back down the nerve there. We see it in measures of vision, and again, as I mentioned, safety. So we'll be in clinic in about 6 months. So we'll see improvements. Does this work as well? Okay. Last thing. Just time. We have some time for questions as well, right? So what can you do? So this sort of is the most common question I get afterwards, so I'm going to just talk about it now. So there was a study about a year ago now done out of Veterans Affairs where they looked at sort of multiple regression. It's an observational study, not experimental. So with all the caveats that comes with that, but they looked at ultimately 8 things they found that correlate with longer lifespan. You could probably guess them, but I'm sure you want to know, so I'll tell you them now. So there's sort of the holy trinity of exercising, and actually not that much. They set the threshold here at like 5 minutes a day. The benefits of exercise, at least from the epidemiological data, really accrue asymptotically. So very small amounts actually are worth a lot. If you're running 2 miles a day, you're basically maxed out. You're pretty far out the curve already. But even like running for 5 minutes a day actually gets you a lot of the steep part of the curve there. So you don't have to exercise much. Encouragement for those of us who need it. Healthy diet, Mediterranean diet, vegetable-based, typically not too much. Sleep well. Then there's the 3 thou shalt nots, I'm sorry to say. Thou shalt not smoke, thou shalt not. It says binge drink, but really any drinking is not good for you. Thou shalt not be addicted to opioids. So that probably generalizes to other drugs, but that's the one they tested in the VA study. And then there's the 2 kind of emotional ones, managing your stress and maintaining good social relationships, both of which also correlate with longer life. Again, it's observational studies, so there was a question on causality, but I think many of us believe these are a good set of things to practice. There's a huge long list of other things you can do, like hyperbaric chambers and supplements, and I'm not going to get into any of that. I will say one of the best validated experimental interventions in humans is Unix actually achieves Sumerian status 130 times more likely than background rate. So have at it if you want. This actually does translate, so if you ablate the germline of a worm, it will live longer. But again, not something I'm suggesting you go do. They'll also stay in your pets for longer. But okay, a little bit of a joke, but it's also serious, right? There's huge value here for each of us personally, and many of us involved there care about it from a personal perspective, but it also has a social value perspective of society as a whole, being able to live longer and healthier. We saw the economic value, and to me, this is as profound as kind of the other big things we've done in history, whether that's 3 examples, there's a long list of others, right? But things that seemed impossible before and are incredibly obvious now. And I believe that'll be the same for this kind of technology. So let me stop there, and but please, we have some time for questions. I'd love to address either scientific questions or just kind of general philosophical ones.
Let's give a warm round of applause. Thank you so much for such a presentation. I think you really set the standard for the Caltech Longevity Club, just with the amount of useful and great information that was shared today. So thank you so much. If anybody has questions, we can start off. I'd love to actually ask them myself. So you mentioned some things that we can do for our personal longevity, but when it comes to contributing to the field, we have so many technological innovators here, people who are so creative and can solve almost any technical problem. What would you recommend to do, and where is the gap in the field that we could help?
Yeah, so there's different answers depending where you're coming from, right? Obviously, a biologist, like there's kind of very clear answers for chemists. Many people who have switched field as well, like come from a physics background, whatever, they move into this, can make great advancements. There's also a huge computational challenge. So as an example, I talked about OSK as kind of 3 transcription factors that we have demonstrated to have some efficacy in animal models. There are 2,000 transcription factors. So if you want to pick a set of those and say, well, which set do I want? I think it's a computationally very hard question. Depending on, you could restrict it and say, I don't want to deliver more than 2, 3, or 4 at a time. Well, it's still 2,000 choose 4 plus 2,000 choose 3 plus 2,000. It's a very large search space problem where computational approaches can be incredibly helpful. There's a similar search space problem in small molecules. There's something like 10 to the 41st possible small molecules that could be drugs. We can probably test 10 to the 7th in the lab, and it's like many orders of magnitude off. So again, computational approaches that help us choose carefully what's the right thing to test. There's a lot of opportunity.
Thank you. And we also had a question actually from one of our participants prepared in advance, which is Shul. Shul, if you'd like to ask your question.
Yeah, I think I have asked that question early on. I do have a different question. So it's sort of like a philosophical question. Basically, I'm curious what's your thoughts on like what is aging? Let me explain. So basically there could be two models. First of all, the first way is that aging there is like a physiological substrate for aging. It could be genes, pathways, modalities, and the other things they read out from these physiological substrates, like causing the disease or malfunction, or the degradation of the different parts of the bodies. So that's one view, and the examples you give, like on mTORC, or the CID study, that kind of, in my perspective, fall into this category. On the other hand, it could be the aging could be an emergent phenomenon where when different parts, body parts fall apart for various reasons, and when disease come into existence, that's what we call aging. And basically, there is no such a central mechanism for aging. And here, like curing the disease, as the example you give, that, in my perspective, can fall into the second category. So I'm just curious what's your thoughts on this.
So there's a lot of evidence of the former, not the latter. So let me give you some evidence. So the first is there are a lot of species that do not age at all. They do not age. And these are typically unicellular species. If you're unicellular, it's very detrimental to age because you go extinct very quickly. Both lines are aging, as with each doubling, right? But they're also all aging, and that line goes extinct. So unicellular organisms that divide symmetrically don't age. Now, a few have actually figured out the trick of they can divide asymmetrically and put the damage into one line, and that line, after 40-something generations, then does die with age. But typically, unicellular organisms don't age. There are also some multicellular organisms that can revert to germ state, so they go from soma back to germ. Things like the hydra that don't age, or that can completely erase their own aging. So there's a lot of evidence comparatively that aging is not this sort of damage model, it is, or things falling apart model, it is a select model. Another way to see that is just in the huge variation you see across species, even closely related ones. So I think the field has, many of us kind of intuitively kind of think of it as, things are falling apart, whatever, but it's actually not the right conceptual basis, and I think it's led the field, I think, a little bit in the wrong direction. So it's a regulated trait, like many other traits, like how tall an organism is, how fast it can run, it's a regulated trait. Because it's regulated, then you can manipulate it.
Yeah, I have a question regarding the eunuchs. Why do you think they live longer? Does this have to do with testosterone or maybe lower metabolism? Just wondering.
So it's a little bit of a weird phenomenon that could have potentially gone either way. If you go back to the theory, way back, it's far left thing, right? So every organism has a limited energy budget. Budget has to be deployed to do work of different kinds. That work can be growth and reproduction, it can be maintenance and repair. Less work on growth and reproduction means more work on maintenance and repair. Now, it's an evolved trait though, so what happens when you actually, so you would say, just thinking mechanistically, if I this out, everything goes here, maintenance and repair is going on. Now, it's a little bit trickier than that because it's an evolved trait. And so things like a drought, a famine, hypoxia, things that have actually really been in the evolutionary history of the organism probably has evolved the right response to. I think becoming a eunuch is like rare in the evolutionary history of most organisms. So it could have it might not necessarily have worked, but it does seem to actually work and kind of hit this trade-off. So I think that's the evolutionary why. I mean, maybe you're asking about how, which is what's mediating the response, and I'm not sure the answer to that one actually, what the signal molecule is.
Okay, thank you. And one more question. I mean, I see a lot of these experts coming out, like Dr. David Sinclair, talking about inhibiting mTOR, lower leucine diets, rapamycin. I just wanted to hear your thoughts on that from first principle basis. And do you think we should be implementing these protocols in order to extend our own lifespans?
So they're good hypotheses, things like mTOR as a target. You know, science is the process of testing your hypotheses in a controlled trial, not saying whether it's true or not. So I can say with a kind of arbitrary p-value that is true for mice. I don't yet have data for humans, but I think there's enough of an inference to say we should do the test and see.
What about monkeys? Has there been any tests?
There has not been a test of something like a pharmacological intervention in monkeys. There was two tests in rhesus monkeys with caloric restriction. Both of them found health benefits. This is actually the two rhesus monkeys you see in the bottom here. So both trials, one was done in Wisconsin and one was done at National Institute of Health. Both trials found health benefits from caloric restriction. Only one of the two found a lifespan benefit. And we can talk about why, but longer-lived organisms are likely to have a shorter period of benefit. So again, if you argue from the evolutionary biology, so you're trying to get another breeding opportunity, another season. If you're a mouse, going from 2 years to 3 years makes a big difference because it's one extra season. You only had 2 to begin with. If you live 50 years already, one extra season probably doesn't make that much of a difference. And so the lifespan benefit should be attenuated in longer-lived species, and that does appear to be what we see. But they did, in one of the studies, saw a small benefit, the other saw none. It was probably kind of close to the noise level for both of them.
Okay, thank you. One last question. I know we asked a lot fast. Many questions. This is the last one. Do you personally fast?
Sort of. I mean, Jewish are fasting Yom Kippur, does that count? I mean, I have occasionally done like a short period of fasting, but it's just tough. I find it hard. So I'm not as good as what I would aspire to.
Hi, thanks so much for coming. I have a bit of maybe esoteric question. So I'm very interested in microbiome for a couple of reasons. One is that bacteria in your body kind of or evolved to interact with certain cells very specifically, so you know, molecules that have very specific effects on very specific pathways. The other reason I'm interested in this is because it's always out there being able to modulate the microbiome. You can feed people probiotics to affect the microbiome, which has an effect on things like chronic inflammation or general health and well-being. So I'm just curious, in the context of aging, what sort of interventions you will employ?
Yeah.
So
It's in the long list of things that you know. There's some evidence that they could work and need to be investigated. Right? So fecal transplants have been shown to have clinical benefit. Not in mice data, but in other parameters that would be associated with health that could lead to lifespan, and certainly health parameters matter anyway. How big the effect is, how sustainable, right? These are things that would have to be explored, but it's for sure on the list of things that people are thinking about. And if you're interested, I would encourage you to follow up on it. There's definitely companies that work in the space, there's labs that work in it.
Great talk. I was wondering about the OSK therapy, and have you ever seen cell identity changes, not like pluripotency? And does it depend on dose, and is that why you have it under inducible promoter?
Okay, so let me go back to this chart. Sorry, it's a little bit painful with all the animations. So if anything, what we've seen with OSK is an increase in immunity, not a decrease. which would be consistent with moving partway back this graph and not all the way back. So for instance, when I showed you those methylation marks, we have the base state methylation, we have the methylation marks after damage and after treatment. What we see is the methylation marks go back to baseline, which would be consistent with the improvement in identity. And those methylation marks in the retinal ganglion cells, which is that particular experiment I showed you, are all related to neurogenesis and axon development, exactly what we would expect around their identity. If we treat OSK in another cell type, in hepatocytes, for example, we see your methylation marks related to liver function. And, so there's some evidence that the way to conceptualize it is you're moving back, but not all the way back. Now, it's an emerging area of science. There is at least a theoretical risk of going too far back, right? Which could be damaging. And so we think it's prudent right now to have it under the inducible system, so we can just turn it off. Obviously hopeful based on the data so far, but that is not an issue. But we're just going into humans for the first time. We want to be careful.
I think that is it for time for today. And if you have any more questions, feel free to send them to the Caltech Longevity Club email or forward to Michael. Let's give another round of applause just for Michael for being here for this presentation. Thank you so much. And thank you everybody for coming as well. Really, you are the next generation of innovators in the longevity field, and your curiosity is the most important thing here. Thank you.
I'm happy to follow up with anyone wants to get connected to folks in the field, that sort of thing. I can't trade and make sure.
And feel free to grab any refreshments on the way out. We have many healthy snacks left and some beautiful talks from our hackathon.
Yeah, it was so great to meet you, by the way. You did a great job. So I can join the podcast. Oh, yeah, and I can join, right? Well, how do we do that? No worries. Yeah, I always try to go to Caltech. Yeah, we'll talk.
Thank you so much for coming.